Huh?
Like you'd be meeting them.
Huh? No, I don't think you have anything to do with no discharge. I don't know who does, but it's like, somebody changed it and, if people don't know, but I mean, this is also, another comment is also, whenever you go to sit down on a microscope, check the temperatures before you load your samples, which someone have found out about.
Me? No, it's never happened to me.
Never happened to you? No. Anyway, yeah, just check settings. I mean, it's not just somebody should always put them back, but it's also just be aware of what the status of whatever you're using is. That's all. And I guess that leaves us up to Kevin. Yeah.
Okay, I won't take up too much of your time. So I'm going to talk about this paper called Cryorium Structure of Gas Vesicles for Buoyancy Controlled Motility. I picked this because I'd never heard of gas vesicles before, so it sounded really interesting. It had a lot of tweets, so maybe it was good. So in any case, it seemed like it was something I could learn something about that I didn't know anything about, so I don't know. Okay, so a couple things about gas vesicles that I just took from Wikipedia. Essentially, these are big proteinaceous hollow spaces filled by gas, and these are permeable to oxygen, nitrogen, hydrogen, carbon dioxide, carbon monoxide, methane, basically all those kind of small gases, only found in bacteria and archaea. So these provide buoyancy and vertical mobility to the organism. So when an organism, I guess, wants to float up, it produces these gas vesicles, and that helps it float up. It's formed solely of proteins, so there's no other macromolecule. And really, it's formed just by one main shell protein called GVPA. And there's a secondary protein called GVPC, which is just an accessory structural protein. These are at least the two that are studied in the manuscript, but the paper also alludes to there being other versions of these proteins in different organisms.
How does the gas get in?
It diffuses, which to me, I guess I can't really picture how it happens, but
Why wouldn't water diffuse?
oh, I don't know. It's a good question.
Something related to hydrophobicity that allows us really non-polar non-polar.
Probably just super hydrophobic on the interior. Is my guess. Okay, so these are not foreign structures that haven't been studied before. People have known about these for a while. So on the top are two, I don't know, electron micrographs looking at bacteria. On the left are kind of the early stages of the formation of these gas vesicles. On the right are kind of the late stages of these vesicles. So initially they start off as some kind of small little nucleated thing, which evolves into a bicone.
I thought it said Bitcoin.
Which evolves into a spindle, and then it gradually grows into a larger cylinder shape. And so I guess in the later stages of growth, these just can occupy a lot of space in the bacteria, and this just helps it float. Okay, so what the...
Can I ask a dumb question?
Sure.
They don't have like any other motility?
Right, so maybe like if it has like a flagella. I don't know. Actually, I didn't look into the species of bacteria that they studied these proteins from, if they have other kinds of mobility. I don't know. It's a good question.
But this is mainly for buoyancy.
Mainly for buoyancy, yeah.
Are these mostly in the salt sea, or is it bacteria, or
Some kind of liquid. I cannot tell you more. Probably salt water.
sometimes if you just look at the name, it says halobacteria. But
Some of them can be filled with helium, so they actually
Yeah, they found out that these bacteria manufacture helium. They what do you call it? Using alchemy to generate helium from.
But you can get helium from rock.
Yeah.
If you're anyway, sorry.
Okay, so the authors wanted to know, so there's been like low resolution studies looking at these vesicles, but these authors wanted to take it to the next level. They wanted to see high resolution details of the gas vesicle shell protein, how it's arranged to actually make the vesicles. They were curious about the mechanisms of gas vesicle biogenesis and also the role of this accessory protein GVPC. Overall, it's kind of just a standard cryo-EM study. There's really not much biochemistry that happens, but the kind of overall structure was sort of interesting. So there's enough material for them to make a paper out of just the cryo-EM stuff. In terms of purifying gas vesicles, so they expressed it in standard E. coli using just IPTG. The kind of interesting thing is that cells that express the gas vesicles naturally float to the top of centrifuge fractions, so they call it the floating fraction. So they could harvest the cells by just centrifuging it, but then taking the floating part on top. They lyse the cells using some detergent lysing, centrifuging, but the gas vesicles always float to the top of the fraction, so it's very easy to work with them and purify them, which,
Can't run them down the column, probably.
no, I don't think so.
You can run the columns, but no.
Okay, so on the left, they're showing a micrograph of a whole E. coli cell expressing recombinant gas vesicle protein. So you can see the interior and even the exterior. There's lots of these cylinders, which are the gas vesicles. They can even identify the kind of pre-gas vesicle nucleated things, the bitcoins, whatever they were. On the right, they're showing their purified cryo-EM samples, showing these kind of rod, spindly shapes with these pointed tips. They noticed that their preps always had different sized vesicles. So you can see the widths of some of these are kind of bigger compared to others. They did some quantification, which I can show in another slide, but here's just kind of a zoomed in version where this one here has a width of 38 nanometers, this one has a width of 53. So it's not a very uniform kind of vesicle shape. So there's probably some ability for this, I don't know, these gas vesicle proteins to kind of just form different sized complexes. Okay, so for structure determination, they don't really make a big point about this, but I assume it was something that they kind of had to struggle with a bit at first. Was they noticed that the larger the gas vesicle was, the more deformed it got as a cryo-EM sample. And so the smaller vesicles actually were the best cryo-EM samples. So on the top here is a micrograph of, I don't know, Anna GV, which tends to form these bigger diameter gas vesicles. And it's supposed to form a kind of perfectly cylindrical shape, but they noticed that there's some deformities along the edges where it kind of, I don't know, it's not a perfect cylinder there. It's kind of bent a bit. But this was not really seen in the smaller gas vesicles.
Or it's flattened differentially along the way. So
Sure.
I actually want to bring up a point about this.
Mm.
When you first got it to this, I finally remembered. So David DeRosier, that I did my thesis with, used to work on these as a hobby. This is years ago before we had a chance. I mean, when you talk about the size of the protein, there wasn't a chance to visualize something that small.
Sure.
You know, actually really solve it. But one of the other problems that comes up in this sample actually is the fact that they have gas in them.
Mm-hmm.
And our imaging and our contrast and stuff is based on the fact that we have our samples in water. So you now have a substantial amount of gas, so you have a different sort of contrast issue also would be a little odd.
Sure.
So the smaller ones would be better actually for that reason also.
Interesting. They've done this little quantification here showing the Anna gas vesicles much bigger compared to these mega gas vesicles, which tend to be smaller. For their cryo-EM studies, they only took gas vesicles, which fell in this very small kind of tail here, like below 40 nanometers in diameter. And so they really only used the super small population of gas vesicles for structure determination. In terms of their cryo-EM workflow, they collected data on Krios. They kind of filtered movies based off which movies had the smallest gas vesicles in them. I don't really know how people do helix reconstructions or whatever, but they did that with Relion and then switched to CryoSpark later on. I guess their sample, since these are kind of heterologous structures, not homogeneous, they could kind of bin their samples by the width of the cylinders, and they only worked on the sample, which was 356.2 angstroms in diameter, and all the other ones they just kind of trashed. But at the end, they had a 3.2 angstrom reconstruction of the helix. So the structure of the gas vesicle, so the protein GVPA, it's very small. It only has 2 alpha helices, 2 beta sheets. It arranges into a secondary structure like this, where there's a hooked N terminus, alpha helix 1, this beta strand which comes down, loops back, and then there's a second helix. A flexible C terminus is desordered. Arranged in the helix, the beta strands are tilted 36 degrees, kind of with respect to the length of the cylinder. Arranged in the helix, the monomers make contact with the next layer of the helix by this N terminus, which is sort of hooked up and makes contact with the beta strand or beta turn, and also with the second alpha helix here. So this kind of zoomed up thing shows the N terminus in blue, and then we have in orange the monomer from the helix above it. Right. They were curious about properties of the structure comparing the interior of the protein to the outside of the protein. So they noticed that basically every amino acid that points towards the center of the gas vesicle is hydrophobic, which would make sense for something that would be filled with gas. So they're showing here the beta strand 1, beta strand 2. On the left side is the gas exposed side of the protein. On the right side is the water exposed. And so on the left side here, we have only isoleucines, alanines, valines going all the way down. Whereas on the right side, there's mostly hydrophilic stuff. So that would maybe explain why there's not much water inside, just because it's totally hydrophobic on the interior.
So that's how you think it, yeah.
Yeah, next slide. Good question. Overall, there was 93 monomers per turn of the helix, approximately. In this class, yes, you're right. Presumably, this is pretty variable.
So they have applied some window to remain in this way. Even going through the previous slide, there is one like that. So yeah. So here in this classification, they have said that. So how they applied this window?
Say it one more time. Sorry.
Because it has been said that they narrow down the entire data to just bring this diameter of the side. So they say that they have done some customized 2D classification something?
So I don't quite know how the 2D classification works, but it does classify, I guess it classified based on the width of the cylinders. And they picked this 356 angstrom one because that one just had the most classes there. Or not classes, but the most particles or picks, yeah, I guess. Okay, so they too were interested in how gas might penetrate into the center of the gas vesicle. So they noticed that between each sort of neighboring subunit, there appeared to be some kind of gap between the alpha 1 helices of the structure and they hypothesized that that gap is wide enough for gases to diffuse through. So they conducted a pore analysis of the gaps. This is a bigger picture of this one here. And the gaps are between 2.4 to 3.8 angstroms, which they believe is enough for gas to diffuse through and make it into the interior of the gas vesicle. They don't have any other kind of experiment to test that, so you can only take it at face value, but that's their hypothesis. Okay, so in doing their classification of their 2D averages, they noticed a small set of particles where it wasn't the same kind of structure. It seemed like it was like two things kind of facing each other, and it turned out that the monomer orientation is such that the beta turn is always facing towards the center of the gas vesicle structure. So, for example, here they've overlaid it with a cartoon of their structure. At the center of the gas vesicle, the beta strands are not oriented in the way I've showed you before, but actually the beta strands are kind of pointing and touching each other from opposing helices. And so this kind of suggests that around the center of the gas vesicle, there's something like a seam, and they predict that that is probably where the gas vesicle grows from.
So how does it not just burst? It's like we're blowing up a balloon and we're gonna like stick a pin in it. So like,
Yeah, I also don't see how it works, but I don't know. So if you'd imagine that,
Imagine gas pressure like anything else reaches a point you can't push it, or something else. I don't know. I mean, there's all these things about what goes on in the cell that makes it really weird.
so they were curious about this mechanism of gas vesicle biogenesis, and kind of playing off the seam that they found at the middle, they kind of assumed that we have one helix on the one side of the cylinder and another helix kind of on the top. The part where the two helices hit and they're opposing, they call this the polarity reversal point, and they assume it's the weakest place in the structure because the way the monomers would kind of touch laterally, there's not much hydrogen bonding compared to how they normally would be. And so they predict that this would be a favorable spot for the kind of structure to slide open and allow the next monomer to attach. Purely hypothetical because they don't have any evidence to really back this up, but it's what they're going with.
It's symmetrical, right? These things. So this process probably mature two monomer comes at the same time. I'm thinking about if only one come, then it would be asymmetrical. Do they always find this seam at the center?
Always at the center. So you're right. I would assume there's two going in at the same time compared to just one. I agree. And they propose this mechanism of biogenesis where there's some kind of nucleating event that starts off with a very small spindle formation, and as it grows, it adopts more of this cylindrical conformation with the seam always at the middle.
Then again, why would, again, I guess you have to close the vesicle, but what's going on at the tip of the column?
Right. So they were also curious about what's happening at the tip. I think they tried to get more out of this, but they just couldn't really get it. So showing a whole gas vesicle here, you can just kind of see that there's not as much stuff happening at the tips. They've done 2D averaging to try to see if they can get out what kind of structure is actually there. But the kind of, there's just, the density kind of just disappears as you get closer and closer to the tip, suggesting that it's just heterogeneous and very flexible at the tip there. They're showing a whole bunch of their 2D averages down here, which I think it looks really cool, but you just can't quite get at what's happening there. They present a model of what they think is happening here. Their ultimate conclusion is that the tips of the gas vesicles are not well resolved, indicating flexible conformations of the monomer there. The author suggests that the N-terminus of the monomers crowd together at the tip. And because the N-terminus is quite flexible, it would just be really, really difficult to get like a really good structure there. So that's kind of the best that they can answer, although it does look like it's not fully closed at the tip. I don't know.
Yeah, certainly can't fit out of that.
Well, yeah, maybe actually what it is, propulsion device shoots gas out of it. No, the other thing you could probably do is do something like, I mean, maybe all of the tips of the protein just aggregate form up lift on the top.
Yeah, maybe. So the last question they wanted to address was the role of this accessory protein, GVPC. So this is as I mentioned before, a structural support protein. It binds on the outside of the gas vesicles and increases the critical collapse pressure of the gas vesicle, which is the amount of pressure you need to apply to actually just burst the vesicle. And what they did was they compared gas vesicles purified with and without the presence of GVPC. So here they're showing 2D averages of the side of a gas vesicle purified with GVPC and without GVPC. The sample with GVPC, they can see clear extra density shown as this white ball, which is not present in the non-GVPC containing fraction. So this is a consistent with the projection of an alpha helix viewed along its helical axis. And that jives pretty well with what people know about this accessory protein, which the structure of this protein is just a big helix. And so they assume that what's happening is that this GVPC protein is just like an extra belt that wraps all the way around the whole helix and provides additional support. The GVPC protein is made up of 5 repeats of the same amino acid sequence. So they, this is like the only biochemistry experiment they did. They found, they did an alignment of a bunch of different species of organisms which have this GVPC accessory protein, aligned that helical repeat, and found that there's a number of completely conserved amino acids, which they assume are the ones that are making contact with the gas vesicle protein. So they present this kind of wheel helix structure, and a number of these completely conserved amino acids are on one face of the helix. So they made mutations of some of these and tested if it changed the critical collapse pressure. So they purified gas vesicles with some of these mutated GVPC proteins. The wild type GVPC protein gives a critical collapse pressure of 4.8 bars. The mutant helices are all equivalent to the negative control, which did not have GVPC, suggesting that these GVPC proteins are not binding to the gas vesicle protein or not providing structural support in the same way. They do some modeling, so they're not able to actually make a model based off the density, because all they can get is this side view looking down the side of the helix, not the side, looking down the helical axis of it. So they did some modeling and docked it in. Include in this paper, they revealed the molecular basis of buoyancy-controlled motility. They talked about arrangement and structure of GVPA, permeability to gases, mechanical stability, biogenesis, and talked about GVPC stabilization. And that's all. Finn.